All right, man. Well, let's jump into this. So,
Yeah.
welcome. Welcome to another Fitomics podcast. So this is I'm Dr. Dan and I am joined with the co-host
Wow.
and CEO of Fitomics. And this is our second podcast. Going to be a little bit more of a short format this time around, where we're going to talk uh, about a particular area of research, and more specifically, one study. We're going to walk through it, and I think this is a interesting topic that uh, deserves a good bit of time to for us to explore and discuss the early research. And so we'll be talking about the research that investigated the intervariability in the skeletal muscle hypertrophy response to resistance training. And so, Cody, this was a big part of your dissertation, or at least a lot of the research that you did during your PhD was kind of trying to dig into some of this, right?
Yeah.
Yeah,
Considerable amount of time for sure.
yeah, um,
Yeah. And piggybacking off of that, yeah, just there's been different words to describe this research. So there's inter-individual variability, there's heterogeneity and responsiveness. So there's these multi-syllable, fancy scientific terms that are really getting at a simple observation that I think most people um, can appreciate that have been working out for any length of time, that some people seem to gain muscle easier than others,
right,
right? In response to the same training program, some people respond better than others. And so it's really trying to understand why at a high
right.
level. Yeah.
Yeah, physiologically, genetically, what's explaining that when you have a group of participants that undergo a standardized resistance training protocol? What's explaining the differences in muscle growth, right?
Right.
And you wrote an extensive review paper on the measurement of skeletal muscle hypertrophy, which would be very fun to talk about in a future talk. Because I think that some of those concepts will tie into this paper. So I have this paper pulled up. And this might be, I think, the first study that looked at some of the potential mechanisms to explain the heterogenetic response to muscle hypertrophy when undergoing a resistance training intervention. And so the title of this paper is Cluster Analysis Tests the Importance of Myogenic Gene Expression During Myofiber Hypertrophy in Humans. And so the first author is Marcus Bamman, who's done a lot of great research,
Yeah.
early
Shout out
research
to Dr.
investigating
Bauman for sure.
this.
Yeah.
Yeah. Um, so, is there anything you want to maybe add about Bauman's work that maybe influenced some of?
Oh man, I mean, legend. He, let's see, so circa 2015, when I started at Auburn, that's when I first met Dr. Bauman. So we went to the UAB Exercises Medicine Symposium when he was at UAB, and really impressive. Just a lot of, I didn't know much about Dr. Bauman's work until I was at Auburn. And that was, like I said, was 2015. This paper was published in 2007. And so he's been at it for a minute. And yeah, has published several really solid studies. Um, but yeah, I mean, this one, it seems that there were a series of papers that were published from the same
Mm-hmm.
intervention. And so around the same time, there was the other paper on myonuclear addition and some satellite cell physiology that seemed to differentiate responsiveness. And building off your point, I believe Dr. Bamman's group was the first to report that,
Right.
and use this k-means cluster analysis to essentially organize participants into groups based on their responsiveness, and then look at different physiological variables within those groups and
Hmm.
see if there's a statistically significant difference, which I know is a controversial term,
The frequentist
between those.
approach.
Yeah, between those groups. And so both the mononuclear addition and satellite cell uh, counts, and then this paper on gene expression, there did seem to be differences between people that responded really well versus those that didn't. But I think I'm jumping ahead. So yeah,
Yeah.
Dr. Bauman, that was like 10 years ago. I met him. Great guy, like really incredible researcher. Dr. Roberts, is a colleague of Dr. Bauman as well,
Hmm.
and published together, and so a lot of respect. This is a really, especially at that time, the this was a very heavy lift. This research project, the
Hmm.
sample size, the 16-week nature of the study, um, like this was a really thorough, well done, in my view, study that required a lot of work. So yeah, shout out to them.
Yeah. Yeah. So we'll definitely, I think, maybe in our next talk, the satellite cell um, paper that was, I think, the 2008 that came after this. We can definitely, I think, dig into that one next for our next talk. But there's maybe some operational definitions that we want to cover. And you talked about one, so gene expression. So maybe could you define that
Sure.
simply?
Yeah. So you've got, and well, the last podcast we did with Dr. Chris Van, he did a nice job of going through the central dogma of molecular biology, which is really DNA. We have our genes, and that those genes are transcribed into RNA. Um, we often measure messenger RNA. There are different kinds of RNA, but you have DNA to RNA, and then the RNA is translated into protein, and that's protein expression. So gene expression is the measurement really of messenger RNA most commonly, in the exercise science domain. And you use PCR, the polymerase chain reaction, to um, do that most commonly. So we're looking at not just is the gene there, but is it being expressed? Is it being transcribed, right, into messenger RNA?
Right. And so transcription and translation, that's an important distinction, I think, especially for this paper. And we'll get into the methods and obviously the results, the juicy part. Um, I don't believe in this paper that they looked at the protein. I believe it was gene
No, not
expression.
not in this one. Yeah,
And that's important, right? Because there can be an upregulation in gene expression, but that doesn't necessarily mean that it ends up getting translated into protein.
that's correct. Yeah,
So that's,
that
I
is a
think, an
very important distinction indeed.
right. Um, so we've defined gene expression. And I think as we go through this, we might come up with some other terms that we can define. I think another thing that's important, perhaps not to insult anybody's intelligence that's listening to this, but mechanical load. That's a term, right? Mechanical tension, mechanical load. So how might we define that
Yeah,
simply?
that's a fair question.
If we're talking
I
about
mean,
resistance training,
yeah, in essence, I think you could say it's a combination of passive and active tension, right, in the fibers. Um, we could belabor that point, but there are forces that are experienced by the muscle cells in response to resisting the movement of our limbs. through external loads or gravity. And the number of times and the relative intensity that we do that influences this whole process.
Well put. And basically, that mechanical tension, that stress, that external force on the muscle tissue is converted into a chemical signal that
Yeah.
results in a signaling cascade
Yeah.
that,
Mechanotransduction is another term you'll see that kind of describes this process. It's transducing a mechanical signal into a biochemical signal.
yeah. And I think they might have used that term somewhere here in this paper. But yeah, so let's dig into it. So I'm just gonna kind of scroll through the introduction here, and I think we have some more operational uh, terms that might be important to discuss. So one of the first things that they talk about in this paper is the expression of IGF-1. And so IGF-1 is the insulin-like growth factor 1. And that there's different isoforms of this, right? There's not just IGF-1. There are variations or splices of this gene. Do
Mhm.
you mind digging into that a little bit? So we have IGF-1. What's the general function of that? Where is it produced? What are maybe, there's two isoforms that they discuss specifically and look at in this paper. So I think the first one that they mention is the IGF-1 isoform EA. And then they have the IGF-1EC or IEC, 1EC, sorry.
Mhm.
So what's
Yeah.
maybe the distinction between those two that would be important for people to know?
Yeah, I mean, and there are others to my understanding. You have these different splice variants, and I mean, we that getting into that whole process would take a little more time, and probably some visual aids would be helpful. I think for our intents and purposes today, to get into like the results and why any of this matters,
Right.
and how we want to leverage the information to practically apply it. The I think the main differentiation here would be there's IGF-1, primarily produced in the liver, it seems, and then uh, MGF, which I believe, Dan, and at this time, I think it's more common to classify the other splice variant. I think is it the EC
Yep. Yeah,
as
that's
yeah,
correct.
mechano growth factor. I think that's a
MGF.
more intuitive name, and that one seems to be primarily produced in the muscle.
Right. And that's in response to the mechanical loading of the skeletal muscle tissue.
Yeah.
And
Now, just real quick, perhaps a little zoom in here could be helpful. We're talking central dogma, DNA to RNA to protein. And as we'll get into in this paper, there did seem to be a difference between the expression. So think of these little mRNA transcripts that are produced from in this process of gene expression, and you can kind of think of a count. So a number of these transcripts that can be translated into protein eventually. Um, the more transcripts, right? The more RNA that's present, that's encoding the information for that growth factor. They did not measure the source code. In this case, they did not measure the DNA level. So the actual gene, its gene expression, and the amount of gene expression for MGF, particularly in this study, seemed to differentiate responsiveness.
right.
Yeah.
Um, so this mechano-growth factor, the expression of it seems to also tie into some of the satellite cell activation. where we
Mm-hmm.
have the, as they say right here in the paper, the myogenic transcription factor family, including things like myogen, myod, myofive, myf6. Um,
Yeah,
and I
and to
believe,
your as we're kind of setting the stage here, so hopefully this isn't like super boring, but a lot of words are poorly defined. We'll throw out these really, like I say, complicated scientific terms of these things, but myogenesis, right? Myo is muscle, genesis is the creation of, right? So there's a process of creating muscle conceptually.
mhm,
Um, and so some of these names are intentional, right? Where it's relating to the process of myogenesis, the creation or regeneration of muscle. And in some sense, right, that's a lot of those pathways are relevant for growth, not just development as we grow up, but and we're in our mothers developing biceps brachii fibers. These signals, the pathways, the myogenic factors are involved to some extent in growth, not just development.
right. Yeah, so this group of proteins that I have highlighted here that I shown on my screen. So these are a group of proteins that regulate muscle formation. So they help with the development, but then even so, muscle cells when they're post-mitotic, these can still play a role, is what you're saying,
Yes. Yeah, and postmitotic meaning that their muscle cells are not going to divide in the classic sense of that term. Yeah.
right? Yeah, and so they have the myod and myofive, I believe. So what we have is we have precursor cells when the muscle is forming during embryogenesis, I believe, correct? And I think myod and the myofive, those commit those precursor cells into becoming a muscle, typically. Might be a way to think of them. And then myogenin and myf6 would direct those to differentiate the cells into a mature muscle fibers. That's during formation. Now their role might be
Hmm.
slightly different in the recovery processes from resistance training. So I think just wanted to define that. We're still on the first page of this study. There's just a lot of things to discuss. Um, so I think going from there, um, again, this was the first lab group that I think reported on the heterogenetic response or the inter subject variability to resistance training.
Hmm.
And um, going to the methods section, why don't we dig into that? Otherwise, we're going to be talking for 2 hours, and this short format podcast is going to be long format. Um, so I think the first thing that we probably want to discuss in the methods is the subjects. Who was sampled in this study? And
Hmm.
so they had 66 participants that were untrained. And this was a wide range of uh, subjects in the sense that we had age groups. We had older individuals that were in the age range, what they say here is 60 to 75 years. Uh, so they had 15 women and 14 men. And then they had a younger group between the ages of 20 and 35. And so for that, they had 16 females, 21 males. So I think that's an important uh, uh, bit of information that we want to hold on to when we're looking at some of this is this was a
Yeah.
wide distribution of age, right?
Yeah.
Um,
And then so 60 to 75, and then the younger, what was the age range?
20 to 35.
Yeah, so young and old adults, older adults.
Well, I'm
Yeah.
glad that we're still classified as younger, being in our
Yeah.
30s. We still fall into that, not middle aged yet.
I don't know. We'll see.
Um, and so then these participants underwent 16 weeks of progressive resistance training. And so they
16
trained,
weeks.
yeah,
Yeah, I mean,
that's
that's
long,
impressive alone. Like a lot of these studies are 8 to 12 weeks. So
right?
not only are we looking at different age groups, we have both males and females represented, and it's a longer duration study than you'll commonly find.
Right.
So those are
Yeah,
some strengths, I would say.
yeah, big strengths. And people are very critical of kind of low-hanging fruit when they read studies in our field that pertain to a resistance training intervention, especially when we're looking at hypertrophy. It's a
Yeah.
common critique that people like to jab at when they're when a new study comes out is the sample size and the length of the study. And the people that are critical of that, I tend to find don't do research, or
Mhm.
at least haven't done it, and don't understand the constraints that we are often faced with, especially in academia where a lot of this research takes place, to conduct a study that is longer than 8 to 12 weeks in particular, and then to
Yep.
recruit a sufficient sample size on top of that, and
Yep.
then to get people to stick with the training intervention
Yep.
and follow it and adhere. And I think they report some of that information, the adherence and how many sessions were actually completed, which is important. Which I believe they reported that between when they classified individuals as non-responders, modest, and high responders, that there was no differences in the adherence to the amount of training
Hmm.
sessions completed. But I just wanted to talk about that because it's just something that I see come up all the time, and it's a fair critique. And we have to interpret studies with that limitation in mind that this is a small, they are typically small sample sizes, even 66 individuals. That's a large sample for this area of research, but that's still a small sample when we're trying to make
Hmm.
inferences to a population of, I don't know, 7 billion people, right? So just wanted to talk about that. And so I guess to go back to the resistance training program, so they train 3 times per week, Monday, Wednesday, Friday, I believe. And they did 3 exercises that included the knee extension, leg press, and squat. But I believe the knee extension was actually the last exercise that they did. I think they talked about the
Hmm.
sequencing of that. So it was most likely squat, leg press, and knee extension, which would be
Oh
kind of
yeah.
a
Set it
typical
up to get
format.
just a gnarly pump on the leg extension at the end. Just
Oh, yeah.
squat it out, leg press it, and then let's go. Hit that leg extension machine.
Yeah. And that's like a classic. I mean, that's just a classic leg workout for quads. Squats, then you
Hmm.
go to a machine, another, I guess you could say, a compound machine movement, and then just, yeah, pumping out the quads with the old leg extension. I This takes me back to, or it makes me think of, so the first bodybuilding. I'm getting off topic here real quick, but when I was in high school, what got me into bodybuilding and I think set me on this trajectory uh, into this field was I bought the Ronnie Coleman book after he won 8 Mr. Olympias, and that was the first legit training program that I followed um, when I was like 15. And I'm just thinking of the images in that book of Ronnie Coleman juiced up, oiled up, and there's a picture of him. I think he's got a cut off flannel in one of them, and just these incredibly short, I don't know, jorts and construction boots, and
Of
he's
course.
just he's just
Hey,
he's got
we're
sunglasses
bodybuilding.
on.
I'm
I
gonna
think
bring
it's
my
like
boots,
dark in a
guys.
gym, and he's just his image is just like quads are just bursting through the shorts. It's just I love that book. It was a solid training program, which we don't have to get into that. I think even for someone that's, enhanced and was a 8-time Mr. Olympia, his training made a lot of sense relative to the current body of uh, evidence in how to structure resistance training. And I guess last point I want to make before we get back to the paper is I anecdotally also was a teenager. So take it for what it is. I went from 125 pounds And then I followed his eating structure and his resistance training program. And over 3 to 4 months, it was like near the end of my sophomore year of high school. And then starting junior year, I went from like 120, 125 pounds to like 165, 170 pounds coming into my junior year.
Right
So
on.
something worked. Um,
Mhm.
but anyways, getting back into it. So they did three leg work, quad specific exercises. And then as far as
And Dan,
the
remind us of the set volume. Was
That's
it
yep. That's
3?
what I'm. Three total sets for each exercise.
So nine sets for the quads conceptually, 3
Hmm.
times a week. So they did 27 sets, working
Yep.
sets for the quads.
They rested 90 seconds, and the training loads were based on their 80% of their baseline one repetition max 1 RM. And
Mhm.
then loads were, I'm just reading from the paper here, loads were incremented in subsequent training sessions when a subject completed 12 repetitions for at least two of the three total sets at a given resistance while maintaining proper form, which I think is really sound way to judge when to increase. So if you have a repetition range and you have three sets, if 2/3 of those or 2 out of 3, you're hitting the 12 reps at least, that's typically a good indicator if you're trying to stick within like an 8 to 12 rep range that you would then maybe want to increase the load for
Mhm.
progressive overload, which might be the most important underpinning um, variable of all when you're trying to get jacked and strong,
Mhm.
is
Yeah,
to make
after
things harder over time.
specificity, yeah, I feel
Right,
like overload is, yeah, we're
right.
tracking.
Um, and I think this was interesting is subjects were taught and reminded to complete both concentric and eccentric phases with control. So they used a 2 second concentric and 2 second eccentric phase. So I'm thinking about that on a squat with like 80% of a 1 RM, which there's a lot of variability in the amount of repetitions that people can complete at a relative percent of their 1 RM. So the amount of repetitions I might complete on a back squat at 80% might be significantly different than what you would complete at your 80%. We've seen that um, throughout a ton of studies that have been published through my dissertation work. We've seen this. Um, but I'm just thinking about this 2 second concentric at 80% of a back squat versus just trying to get the freaking weight up. That had to
Mhm.
be brutal if they were really adhering to that. And there is difficulties in that. Again, when you report something like a tempo, sometimes, as you know, when you do a resistance training intervention for a study, you have variables that you're trying to control. Getting people to go to failure, getting people to follow a specific tempo doesn't always work out perfectly every time. You have some subjects or participants that execute things perfectly, and then some that don't. So there was probably, if I had to speculate, some variability in the tempo here. I think a 2 second concentric deliberately trying to execute that on a back squat in particular at 80% would be difficult, especially for a very strong individual. Um, but moving on, I digress.
That's a yeah, fair take, and I guess another perspective on it would be some of that variability in tempo, perhaps eccentrically or concentrically, you would expect that to kinda wash out in 66
Right,
participants,
right.
right?
Not really a critique, just more of an observation or something that,
Yeah, consideration, yeah.
yeah, which tempo seems to be not a major contributing factor with
Mhm.
respect to skeletal muscle hypertrophy.
Right.
Um, so then the next section of the methods is the muscle biopsy and tissue preparation. So briefly, um, Cody, what is a muscle biopsy? How do we do it? What's your 30-second take on that or
Yeah,
explanation?
we get a really dull knife and turn on Lincoln Park, and we cut all the way from the patella up to the iliac crest and separate the flesh and with our bare hands that have been unsanitized, we just grab the vastus lateralis and rip it out. And that's how we do a muscle biopsy. At least that's how we did it at Auburn. And the IRB didn't know that. Um, so I'm joking, everyone. It's a joke.
And the
So yeah,
IRB is, just for people that are wondering what that acronym stands for, it's the Institutional Review Board. So it's a committee that approves the ethics of the study, um,
theoretically, yes,
which we could
yes.
certainly talk about the IRB process, but I don't want to get upset. But
Mm.
so the next thing, so after we
So
do a
yeah,
muscle
the
biopsy,
biopsy.
yeah, so
Yeah,
there's typically,
there's a 9 gauge needle, and yeah, it's got an opening, and there's another part with, yeah, without getting too specific, you, you carefully sanitize the area. There's local anesthetic, and then you make a pilot incision and you insert the biopsy needle, and it's connected to suction, and we clip a small, like, pencil eraser sized piece of muscle. Sometimes it's a little larger. There are also microbiopsy techniques for even smaller samples, but this one used the Bergstrom technique, I believe. And so, we get this, like I say, pencil eraser sized piece of muscle, and yeah, that's how we would do a biopsy.
so
And
not a
then
dull
you
knife.
know, not a dull knife in this case, yeah.
So once we have the tissue sample, there's some other steps, I think, to separate the tissue,
Mhm.
the skeletal muscle tissue, which I've
Yes.
watched you do at Auburn when I was a master student. I was your master student at LaGrange College. Shout out to LaGrange. Good
Mhm.
times. And so then one of the things was RNA that we talked about that. So there's total RNA isolation. So
Yep.
what are kind of like the basic steps or important takes for that?
Well, yeah, I mean, there's several steps, but I think, again, for this overview, because the methods section of this paper alone, like, if you really wanted to go through every single thing that they did, there's quite a few steps of processing here. But really, what we're trying to do with a portion of that tissue, we often separate it, flash freeze a portion for immunohistochemical staining. So it's going to get sliced and mounted onto a microscope slide. That's part of the tissue is reserved for that. Sometimes we'll reserve another section for protein analysis, and then yet another section of that sample for RNA extraction. So what we're doing there is extracting the RNA, and so you use different, chemical agents to do that. There's precipitation steps and centrifugation, but I think for this, just understanding that after we get that tissue, we're going to extract RNA from it,
Hmm. I've
and we
done,
yeah.
yeah, I was gonna say I've attempted to do some RNA isolation before, failed, but I haven't done it from using uh, human skeletal muscle tissue. But it is that whole process of isolating RNA, then going into um, PCR, or I think they used RT-PCR, which is real time polymerase chain reaction. Um, that's it's a lot of work, especially depending if you're doing things in duplicates or triplicates,
Right.
um, and then the amount of different uh, groups.
Yeah.
It can be a full day event over
Oh, yeah,
multiple days.
multiple days, yeah.
Yeah, I think it was 40 hours that I had put in attempting this over like 3 days, being
Mhm.
in a biochem lab until midnight. Um, it's a lot of work. Anyways, I just noticed that table one has the resistance training program summary. So they show by group non-responders, modest responders, and extreme responders. And so they show squat, leg press, knee extension. They have the average amount of repetitions completed, the average percent of 1RM, and the adherence. So they do report this in a table. The adherence to the resistance training protocol was around 90%, give or take. The average training intensity for squat was looks as low as like 73% up to like 77% for the squat, and then 77 to 80% for leg press. And I believe that they mentioned because of the fatigue um, that participants had by the time they got to the knee extension, that was a lower percent of their 1RM as they report here. Was typically under 70%. So anywhere from 62 to 68%. And so I think when we before we get into the PCR real quick, the subjects, we had 66 subjects. I think one thing that's important, and as they show in this table, is the distribution when they did their clustering, is that there was 17 participants that were classified as non-responders, 32 as modest responders, and 17 as extreme responders. We'll come back to that. I just wanted to um, make sure that that was stated. Um, so circling back to the RNA isolation, once we've isolated the RNA, then we would go into RT-PCR. And this is when we're trying to amplify a gene to look at gene expression, right?
Yep.
And the specific primers that they used here, the genes that they were looking at were the MGF, so the mechano-growth factor, IGF-1, which I believe they looked at the both isoforms, and myogenin and myod mRNAs. Um, so following, is there anything that you would want to add to the RT-PCR for this talk?
I mean, I think yeah, for again our intents and purposes today, that yeah, it's a cyclical process of amplifying transcript abundance. So you run these cycles on a PCR machine that duplicates copy numbers every cycle conceptually. That's just kind of a high level overview.
Yeah.
So you can amplify that signal and better detect and differentiate expression patterns. And yeah, the primer design is another step that we're looking at very specific um, orders of these, like all these genes, right, have a nucleotide sequence,
Mm-hmm.
and it's very specific, and you have primers basically that help to identify that specific sequence of interest. And on PubMed, the NCBI, like we had a whole process of primer design. So you can go and you can pull that information and design primers, and then that's how
Mm-hmm.
you identify the specific transcripts. So it's a highly specific technique, and this goes back decades.
Yeah.
This is one of the most common molecular biology techniques. So it's a very well respected technique. So I think we can have confidence in the information here.
Yeah. And so then following RT-PCR, they did the immunofluorescence microscopy. I can't say this. Microscopy. Oh boy, stumbling over my words here. Um, so kind of a brief explanation of that. Otherwise, I could just read it from the methods here. But have you
Yeah,
used this
man. I
before? I'm
mean,
sure.
yeah, a lot of microscopy work at Auburn. Um, so yeah, we take that tissue, we slice it on a cryotome into very thin, like micron thin slices. You put it on a microscope slide, spit on it, and then put it under a microscope. No, I'm kidding. So we're gonna put certain solutions and eventually antibodies that help us to identify the structures within the cell, and particularly the membrane is an important portion of that cross section of tissue to identify, because that's the border of the muscle fiber, right? So if you kind of think of longitudinally in 3 dimensions, and we're turning a muscle fiber, if it's facing you and you're looking down a tube conceptually, or a series of tubes, and in this one they actually I believe it was like 150 to 300 fibers per cross section, which is a nice sample. And so you're looking at somewhere between 150 and 300 fibers per cross section from the vastus lateralis muscle. And so when we stain it, you can see those membranes. And with a microscope, so using light microscopy, fluorescence, and the right kind of antibodies and secondary antibodies, you can have different colors in that cross section that allow you to differentiate the different structures or proteins of interest. And the membranes, particularly, are important to calculate cross sectional area, right? The size of the fibers. And
Mm.
that's what was used in this case to differentiate the amount of muscle growth that occurred in a participant. So if you've got like a fiber this size, right, at baseline, according to the cross section, let's say it was 3,000 microns squared. Well, and then it gets this big. So now it's 5,000 or 6,000. Well, that's one definition of hypertrophy, an increase in cross sectional area. And the magnitude of the increase in this case was used to determine if someone was an extreme, moderate, or non-responder.
Yeah, and I think that ties in well. So if we look at the statistical analysis, is they used what's called a k-means cluster, and so this is a statistical method where they group data into clusters based on shared characteristics. So each group is more or less homogeneous, and that's how they classified, like you said, the non-responders, modest, and high responders was based on that mean change in the cross-sectional area of the myofiber. And I
Mhm.
believe they report type 1 and type 2. Um, but I think in this figure, so I'm looking at figure 1, and I think that this is, now we're getting into the results section. So this is the juicy part. And I think the methods, we spent a lot of time talking about it, but I think it's important, is understanding the methodology that went into deriving the results, which helps us interpret it more appropriately,
Right.
I think.
Yep.
Um, so now I'm looking at figure 1, and this on the y-axis is the change in mean myofiber cross-sectional area, which is measured in the unit of micrometers squared. And then along the x-axis is we have our three groups, which we see 1, 2, 3, which one is the non-responders, 2 is the modest, and then 3 would be the extreme or high responders. And this is what I really appreciate from studies, not just with things that are studies investigating skeletal muscle hypertrophy, but pretty much any studies when they show the individual data points like this versus just like a bar plot or
Mhm.
something like that, right? Or where they just plot the mean data point with the error bars, like standard deviation or
Mhm.
the standard error. Because I think that there's a lot of interesting stuff that we can derive just from looking at this single figure,
Yeah.
right? And so the non-responders, if we were just to look at it's a triangle, it's filled in and it has the error bars, which would be the mean. It's right around 0. So there wasn't really any change just looking at this figure in the non-responders. And I think the distribution is something that we should talk about here, is how many males, females, and the age group
Yeah.
for each of these would be important.
Yep.
Um, and so what they say here in the results is the focus of this work was on differences in gene expression across responder clusters, irrespective of age or sex. So their analysis didn't necessarily account for age or sex, but it kind of did with the clustering. But they say, however, it is noteworthy that the non-responders contained only one young male compared with 38% older women and men. By contrast, young men clearly dominated the extreme cluster, as 43% of the young men were in extreme compared with only 18% of the remaining subjects. Approximately 50% of each age sex cohort was clustered as modest. So there was more young men that made up the extreme cluster, but there was males and females that fell into each of the clusters, and by age group also fell into each cluster. So there were high responders that were older. And I think that's interesting, is it wasn't just like all of the young 20 to 35-year-old males were all extreme responders, right? That this
Right.
the age group and the sex was distributed across the clusters. And so 50%, when we go back to figure one, about 50% of the participants in this study were classified as modest. And about 25% were non-responders and 25% were the extreme responders. And so I think if we're making some inferences here to the uh, broader population,
Mm-hmm.
is, of course, caution. There's some caution that we want to do with that with a small sample size. But if we were just to take this study and this sample size and make some inferences, is the modest group experienced some pretty, uh, I think, from the research that was published previously from other studies, that this was considered normal or expected in the CSA change of the myofibers. And so about like 50% of these participants, and if we're inferring to the population, about 50% of people that resistance train
Mm-hmm.
will experience a modest amount of growth, which I
Right.
think if you go to the gym and you see the regulars, you could probably see that, you know, uh, play out in real life, where you have regulars that go to the gym, they work out, looks like they work out, but then you have people that look like they're on something, and maybe they are. And those would be the
Mm-hmm.
extreme responders. And they classified this group as not being normal relative to, at that time, the current body of evidence, that these were outside the norms that had been previously reported. And so they grew significantly more. And that was about 25% of the participants in this uh, study. And then the non responders, what's really interesting, and this is something that I've picked up on when going through quite a few studies looking at muscle hypertrophy that have at least measured it, is when they plot individual data points like this, sometimes you'll see data points where people actually get smaller, at least in that measurement, where there's a decrease below baseline. And we can see that here in the non responders. And I think that that's just, it's really curious, right? Like they
Yeah.
got worse over 16
Right.
weeks, at least in this measurement,
Mm-hmm.
right? And there's a lot of limitations with that, but some people actually their measurement got smaller, and I think maybe tying back into the method section with the muscle biopsy, there's a lot of limitations with that, right? Because like you said, we're extracting this tissue at baseline, pre resistance training intervention. That tissue's gone, right?
Mm-hmm.
Post intervention, we or they would go back into that insertion point. They measure it, standardize where they take the measurement from. They go back in and pull out more muscle tissue, but it's not the same tissue, right?
Yeah, I mean theoretically, and yeah, I mean even standardizing where the biopsy is collected, it's certainly possible. if not intentional, depending on the nature of the study. Of course, I didn't take these biopsies, so I don't know exactly where the site occurred, but it may not actually be, the same millimeter placement
Hmm.
on the vas lateralis. So yeah, you might be sampling, and probably so, other fibers, um, proximal to the original biopsy site.
Yeah. So there's just some inherent limitations with this. Is again, they only looked at as far as the hypertrophy measure was the muscle fiber itself. It wasn't a gross measure using something
Exactly.
like
Yeah, I think
MRI or ultrasound.
that's one of the main points, Dan. Yeah. Is like the variability in the technique itself. Well, first off, again, the pencil eraser sized piece of muscle from one muscle in the body.
Hmm.
Okay. Like think about that, right? So this is not a whole body muscle mass assessment.
Right.
It's 150 to 300 fibers in one muscle.
Hmm.
Now, um, yeah, like a PQCT or MRI, um, heck, even like an ultrasound. You have levels of measurement and volume of tissue there that different studies will use, and that's, you talked about the review article. That was one of the key intentions of that work, is to like make sure that we understand the different kinds of measurement. When we talk about hypertrophy, we look at it as an outcome variable, but it's a construct and you can measure it differently. And how you measure it makes a difference in the findings, and they don't always agree. So, like in
Hmm.
this study, it's possible that if they had used something like a PQCT, which is a form of computed tomography where it's peripheral quantitative, so you can measure the volume of muscle in a limb,
Hmm.
the whole volume, or something like MRI, where you're getting whole muscle volumes. Maybe the individuals that were classified as non-responders would have been moderate responders, or I'm not sure, it's possible, but maybe they were extreme responders
Hmm.
with another measurement. So we don't know that, in this study, but we have seen that the measurements, if you do multiple levels of measurement, they don't always agree.
Right.
Which, like, you use the word curious, and yeah, that's one word to describe it. Confusing is another one.
Hmm.
Um, but yeah, that is an important distinction here.
Yeah. Yeah. I think there was a more recent paper out of Dr. Mike Roberts' lab with Bradley Rupel as the first author that ran a bunch of uh, correlations between all of
Hmm.
the measures, and they weren't really correlated.
Hmm.
um, they didn't explain a lot of variance in one another. But there was also
Yep.
older research long before that that had done some basic correlations. That I think when we talk with Alex about the motor unit stuff, in the paper, the Pope paper, that they talk about some old research from like the 70s or 80s that was looking at the CSA of type 1 and type 2 fibers relative to muscle CSA. Um,
Hmm.
so yeah, I think that's an important point. Is
Yeah.
at the end of the day, we want to look like Ronnie Coleman did in his bodybuilding book. Uh, and the question is, does this change in the mean myofiber CSA translate into an aesthetic change? Really, I think that's what we're really after. Is we do these measures, but is it aesthetically? Do we look like an 8-time Mr. Olympia Ronnie Coleman after a resistance training intervention, right? Or at least do we have some sort of visually noticeable change in hypertrophy? Um,
Hmm.
so yeah, I think that's a really, really good point. Um, but they did do some other measures that I think support maybe these findings with the from the PCR with the gene expression and stuff like that. And they did 1RM testing, which was also very interesting, which I definitely want to talk about. So I'm scrolling down. I'm going to figure two, and so they have bar plots here. Again, differentiating the non-modest and extreme responders. And the top plot is the mechano-growth factor, MGF, mRNAs. And at baseline, there wasn't any differences statistically that they reported. But there was an acute difference in the level of expression in MGF in both the modest and extreme responders, which was a 24-hour acute response to the first resistance training session. And then 16 weeks later, there was an even greater increase. I don't know if it was different than the acute, but there was a um, even greater uh, expression of MGF in the extreme responders relative to baseline. And that was also
Yeah.
observed in the uh, modest group. Now, what's interesting here that I've been kind of scratching my head about is when you just look at it, the eyeball test, as
Mm-hmm.
um, you've told me, start with the
Mm-hmm.
eyeball test. When you just look at this, if we didn't have these annotations above it, my first inclination would be that the non-responders also would have had a significant increase in MGF if we just look at it, right? If you look at the baseline to 16 weeks post,
Mm-hmm.
it looks like it's increased, but
Yeah.
based on their ANOVA and
Mm-hmm.
likely post hoc test, they didn't find anything significant, even though visually it looks like there was an increase and there could have been a wide amount of variability in that group in the response. I believe those error bars are the standard error versus standard deviation. So maybe the standard
Yeah. Those,
deviation was, yeah,
yeah. Those standard deviation bars would be
pretty wide,
large,
which they would probably intersect or cross over from baseline if we were
probably.
to probably, right? Um,
Yeah.
to where they wouldn't be viewed as different. So that would probably be the explanation with that. Um, versus that variability was much tighter in the modest and the extreme responders. And then there was an upregulation in the IGF-1 as well, but that expression was significant for all groups,
Yeah.
or at least it was different from the previous time point. That's little asterisk. That's how they define that in the figure key. Different from previous time point within group. So I would assume that that was at least different from baseline or acute. Um, and I think one interpretation that they had was that IGF-1 wasn't necessarily correlated based on this to the expected change in myofiber hypertrophy,
Yeah.
even though it was statistically significant.
Yeah. Like the expression of IGF-1 messenger RNA increased relative to baseline in all groups,
Mm-hmm.
but that didn't seem to explain the growth response.
Correct.
Yeah.
So I think that's interesting.
Yeah. On the other hand, the NGF did.
Yes.
Mhm. The mechano-growth factor. And I guess summarizing briefly, you were saying that there's a qualitative using the eye test. Acutely, it looks like MGF mRNA expression increased in all subjects
Yes.
and after 16 weeks. So transcript abundance was higher. It just seemed that the moderate and particularly the extreme responders displayed a larger increase in mechano-growth factor expression.
Correct. And like we were talking about, there was probably less variability in that group.
Yeah.
And I believe in the discussion, they speculate some of this, um, that its potential explanation is that the non-responders, for unknown reasons, are less sensitive. The receptors that would perceive this mechanical load and then result in a signaling cascade, they might be just less sensitive.
Well,
Um,
and you're touching on something, just connecting some dots here. In the extreme responder cluster, there were quite a few younger adults.
correct.
Yeah.
Yes. Yeah, that's a good.
Yeah.
Yeah.
Yeah. So, and we know that from other lines of research since this study, this phenomenon of anabolic resistance, and that's
Mm-hmm.
a multivariate thing. This may be one thing, but it does also seem to be mediated by even a sensitivity or lack thereof to amino acids. So it's not just the mechanical signal, perhaps, right? It's the actual amino acid based signaling. When
Mm-hmm.
we ingest protein, it seems like older individuals have a little bit more of a resistance to increase muscle protein synthesis in response to the same amount of protein ingestion or essential amino acid availability.
Mm-hmm. Yeah,
Yeah.
that's a really good point. But there were older individuals that were classified as high responders,
Hmm.
which again is a little bit of a head scratcher when you consider the anabolic resistance. Um,
Hmm.
but I guess so. Moving on, looking at the table two, where they report the, uh, I guess as we would classify the muscle fiber types into three categories, where we have type one, type two, and then they have the distribution in a percentage of that. Basically, the change, how these muscle fibers can shift, is a phenomenon that's been reported and talked about. Um, and I guess without getting too far into the weeds with that, is you have your type 1 muscle fibers, which are your aerobic or oxidative muscle fibers. Think more endurance based with that. And then you have your, on the other, that would be on one end of the spectrum. And then on the complete opposite end of the spectrum is you would have your type 2X, which are your high force, high fatiguing muscle fibers that are also, they're very sensitive to muscle contractions. Um, and then somewhere in the middle is kind of a hybrid of the two, where you'd have your type 2As, which are a blend of oxidative and glycolytic fibers. Um, and correct me if I'm wrong, but typically when we undergo exercise, such as resistance training, is we might see a shift in this continuum. Maybe the type 1s shift a little bit more to express type 2A qualities, and the type 2X, because they're so sensitive and through certain signals like calcium, might shift from a type 2X to a type 2A.
Hmm.
And people have, especially in early research, it was reported that the type 2 muscle fibers were the ones that experienced the greatest amount of growth, and maybe the only muscle fibers that experience growth, I think was reported in the early literature. There was at least, I think, one study that reported that. Um, but I don't think that the current body of evidence supports that. It does seem that the type 1 muscle fibers do hypertrophy, which we can see, um, at least within the modest and extreme responders, that they look like they increased, the type 1s. especially from the extreme responders, with the measurement of like 4,131 um, micrometers to almost 6,000 is what the CSA increase was from type 1 within type 1 muscle fibers. And then if we
Yeah,
look at
so
the
that's
type,
like a 50% increase in cross-sectional area.
right, of the type 1 muscle fibers, so they do grow. They are responsive based on this data to mechanical loading. Um,
Mhm.
and the type 2 muscle fibers also grew quite significantly,
Yep.
maybe even to a greater extent than the type 1s. Again, if we're just looking at the raw data here, like in the extreme responders, it went from 4,200 micrometers to 7,000 was the group average.
Yeah, that's like a seven that the average is like a 75% increase.
Yeah, yeah, that's crazy. Um, and then just the percent distribution of these muscle fibers. Um, the type 1, the percentage distribution didn't really change. But what does seem to change quite a bit was the type 2X, like we were talking about, when they're extremely sensitive, and you tend to see a greater expression of, and correct me if I'm wrong, Cody, the myosin heavy chain type 2X, if we were to measure that, would probably be greater in sedentary populations versus active. And some of that's due to the muscle contractions and the repeated exposure to things like calcium and other signaling molecules like what is it, calcineurin, calmodulin.
Mhm.
Those things can result in a shift of the type 2, especially the type 2Xs, towards that type 2A and type 1
Mhm.
phenotype, which has interesting implications for strength and conditioning coaches with the timing of periodization and trying to maximize these type 2X muscle fibers. They would be very high force producing, high velocity, right? Very powerful muscle fibers, but not getting into the weeds there. I think what's interesting is it almost went to zero, the percentage distribution of the type 2Xs. It was like hanging around 12 to 15% for all three clusters, and then it goes to like 1 to 2% after 16 weeks. So I think that that's
Yep.
interesting. So there was
Yeah,
definitely a
yeah.
shift in the type
And
2A.
yeah, for further reading, Daniel Plotkin has an excellent
Hmm.
review on this
Yep.
that, I was able to be a part of, and it does a nice job of summarizing that literature. And we used to think that fiber types wouldn't really convert or shift, and I think now we know that they do
Yeah.
based on how you train and other factors. But training is a powerful tool to
Hmm.
manipulate the characteristics of the muscle tissue.
Right. All right. And so moving on to figure 3 briefly, we have the myogenin mRNAs on the top figure, figure A, and then we have MyoD mRNAs for figure B. And I guess nothing was found that was statistically significant in the MyoD mRNAs. And
Mhm.
kind of circling back again, I think the MyoD and MyO5 are during muscle formation commit precursor cells into becoming a muscle. But the myogenin mRNAs were found to be statistically significantly expressed in the modest and extreme responders. So that could also be an important factor, the myogenin, um, in explaining some of this variability in the response to resistance training with respect to changes in uh, myofiber CSA. Um, but what's really interesting that uh, I like is figure 4 with the 1RM testing, because there's a hot topic, and Alex, who we'll be talking with about some motor unit stuff, this was a big part of his dissertation and his current area of interest with research is what is explaining the increase in strength. And is it muscle hypertrophy? Is it neuromuscular? Is it a combination of both? Um, but this figure, I think, makes a really convincing argument that larger muscle fibers
Mm.
are necessary for the later increases in 1RM strength that we experience. And so, what I mean by later is classically what we have said in the exercise science community is the early stages of resistance training, the first 6 weeks, are predominantly neural adaptations. So, don't have to get into the weeds with all of that, but it's predominantly neural. And it's, as far as the way that we have measured hypertrophy, is at least within the first 4 weeks, we don't detect changes in muscle hypertrophy that weren't attributed to something like edema, right? I think DeFreitas had a paper on that, like the early weeks of resistance training, measuring some of that. But here at 8 weeks, so 2 weeks post 6 weeks of neural adaptations, there was a continual increase in 1RM strength. And we see that that increase in the 1RM strength is even greater in the modest and extreme responders. And so if neural adaptations are little to none post 6 weeks, then what is explaining this continual increase in 1RM strength? Well, based on the data that they've reported here, is that the muscle fiber CSA got bigger.
Right.
uh, so if it's not neural, it has to be something else that's explaining this increase.
Yeah.
And what else could it be other than increasing the size of the contractile proteins that are mechanically moving the load on your back when you're doing a barbell back squat?
Yeah, makes sense to me.
Makes a lot of sense.
Yeah, so like the actual myosin and actin proteins that interact to bring the Z lines together in the sarcomere to contract the muscle, that interaction, the cross bridge that's formed, is really kind of at the heart of force production in the muscle. So if you have more of those, right, the force one is able to produce would likely be greater, which strength, I think, simply defined as the ability to produce force. And so, yeah, I mean, that makes sense that we would see that.
Right. And they discuss this in the discussion section. Um, but I think this study is very strong evidence in support of that argument, which there is a group of individuals that would argue that there isn't great evidence supporting the hypothesis that larger muscles are required to lift heavier weight, and that it's larger muscles are just kind of a byproduct of progressive resistance training. But I think this is interesting. The non-responders still seemed to have a small increase looking at the average
Hmm.
from 8 weeks to 16 weeks. They did have, and again, if we looked at the first figure with all the data points, baseline was at 0. There were people, some data points that were above 0. So some individuals still experienced some myofibular hypertrophy. Um, so they increased its strength. I guess all that to say is I think that there's just good evidence here from this study to support that larger muscle fibers likely contribute to increased force production and strength. Um,
Yep.
so that would
Yeah.
lead us. Yeah, go ahead.
And yeah, I think one thing to conclude for now, because yeah, we could definitely, and this is the first of I think a series of cool papers that have differentiated responsiveness that we'll be digging into. So one of the things to point out now, this goes back to some of Frank Booth's work. You know, legend in exercise physiology. Really, I would say, in some sense, the godfather of molecular exercise physiology. And Dr. Roberts, did his postdoc research with Dr. Booth, and Dr. Booth is still getting after it. At the University of Missouri, and doing some really cool work even now. And so um, there's this idea. Dr. Stone used to talk about this at ETSU as well regarding the molecular processes that influence muscle growth, and it's this idea of an mRNA pool and threshold. So, like to your point, the we often when we look at a time course of muscle growth, if you've got and we have, you know, um, let's just look at the pre-post data in this study. We don't have, I'm not aware of like a study that did weekly biopsies for like 16 weeks to understand that time course of change,
Mm-hmm.
because it's unlikely to be linear. I'm not saying it's exponential, but it's perhaps curvilinear. And the changes in muscle size are often too small to detect in the first few weeks of a
Right.
training intervention. And so, yeah, we often interpret that as, well, there are neural adaptations that are happening, like increased motor unit recruitment and rate coding and synchronization. We're getting
Mm-hmm.
better at activating the muscles to produce force. And so those changes influence getting stronger early on. And then as we kind of, let's say, saturate that ability, so we've learned how to contract our quads during a knee extension after doing it for a few weeks, got it. Well, now we start to accumulate uh, proteins, and we start to see increase in size that further contributes to increases in strength. Well, this mRNA pool, if you think about these transcripts, this study measured acute gene expression and saw an increase in mechano-growth factor RNA and IGF-1 RNA, right? So those transcripts don't just go away, right? We talked about transcription and translation. So those transcripts have to be translated. We also know that ribosome content goes up over time. Those are the machines that translate the RNAs. So perhaps there needs to be this pool of mRNA, a threshold in some sense, where we accumulate these RNA transcripts, and then eventually there's so much of them present and ribosomes that there's more spatial interaction between the transcripts and the ribosomes. They're getting closer together in some sense,
Mm.
right? And so then that really starts to ramp up translation. And so there's a delayed, hypertrophic response when you continue training, and you got to stick with it, right? You got to continue past that first few weeks to keep providing that unambiguous signal of what you want the cells to do. And I think that informs programming, right?
Absolutely.
Like not changing your training programming too much on a regular basis to saturate that adaptive response, across time. Um, and it would have been cool if they would have just kept doing the same training program
Mm-hmm.
for another 16 weeks. Would the clusters have, plateaued, right?
Mm-hmm.
And then seeing when that happens could inform program design and manipulation when we need to switch the emphasis and perhaps de-emphasize those movements and prioritizing those muscle groups and prioritizing others, instead. So just a little insight on that.
Yeah. Well, I think that's a good insight to end this talk. Um, yeah. Well, why don't we end it there? And we will be doing a follow up to some of Bauman's work, and then we'll continue to work our way historically to more recent evidence and literature on this topic. Um, this was great. All
Right
right.
on.
Well, appreciate you listening, whoever you are, and make sure if you're watching this on YouTube or Spotify, Apple Podcast, to subscribe. In the comments section of YouTube, let us know if you have any questions. Thanks for listening, and peace.
Yep. God bless.